Electricity supply has been cut off from your area, from your house. You have no electricity around you or even in your neighbors. So most people in my country would relate to this scenario.
You get it? If you don't get it, forget about it.
But for those of you who cannot relate, try to imagine it. Try to imagine how life will feel like, especially in this age and time where most of our activities, even our work, is somehow related or somehow tied to the use of electricity. So how would it feel like? Usually slow, low energy, boring, and low motivation. So somehow your cells and your body in general will feel the same when their main power supply is cut off. Now, where does electricity come from? Our engineers say that it is generated in a power plant which is usually made of a dam that has been created or a dam that is natural that is able to generate electricity from water. So basically, a power plant. So the body also has its power plant, which is the mitochondria. It is found in every cell of the body. The mitochondria powers the cell with every energy that the cell needs to carry out its function. So it does that through a process called the electron transport chain or oxidative phosphorylation. The electron transport chain, a chain which means that the events happen successively. So in a chain of events, the previous event or the previous molecule in the chain would lead to the next molecule or the next event in that chain. So in the electron transport chain, electrons are moved from one component of the chain to the next component of the chain. So going back to our electricity supply, the power plant, we can also try to imagine what happens there, the chain of activities that happen for water to be converted into electrical energy. Probably water falling from the dam, passing through or flowing through pipes, what they call penstocks or whatever, and maybe eventually the turbine spinning the water and then eventually producing electricity. So it's a chain of events that will probably be happening there. And we're going to be looking at the chain of events that happen in the cell's mitochondria for us to be able to have energy that powers the cells and powers our whole body. So, as usual, we're going to be making it as simple and as relatable as possible, which is what we do with all of our videos here on Curious Molecules. So if you're new here, you're very much welcome. And if you're returning, please do not forget to subscribe, to like, and to share the video too. Okay, so we're getting straight into it. We're talking about the electron transport chain today. The electron transport chain, also called oxidative phosphorylation, which is the cell's power plant, as we have earlier introduced, the cell's power plant. So we get right into the components of the electron transport chain. Remember in our past videos of introducing metabolism or metabolic pathways of carbohydrates, we always talk about what's involved in that pathway. And we usually most times start with where it happens. Where does this pathway happen? In the inner mitochondrial membrane. That's where electron transport happens. Looking at the structure of the mitochondria, we can see that, or the mitochondrium for one, we can see that it has an outer covering, which is the outer membrane. But the structure within it also has its membrane, which is the matrix. The matrix has a membrane, and that membrane makes it the inner mitochondrial membrane, the covering of the matrix. And then the matrix is basically the insides of the mitochondria, or the components inside make up the matrix. And the mitochondria, of course, has its own DNA, which is found inside, and all other molecules that it needs to carry out its function. So basically, this is how the mitochondria looks like in cell biology. And then the space, since it has two membranes, the space between the membranes is called the intermembrane space. Take note of the intermembrane space, the outer membrane, the inner membrane, and the matrix, because we're going to be talking a lot about them in the process of electron transport. All right. In other pathways, since most of them were made up of 10 steps, a lot of steps, we didn't maybe list the components of every step. We just went into the reactions that occur. But here we're going to be listing the components because they are few, although they are complexes, and they are protein complexes. We have complex 1, complex 2, complex 3, and complex 4. Complex 1 is what we have seen before, something similar to what we have seen before. NADH or NAD, oxidoreductase. Complex 2 is something we have also seen before in the TCA, succinate reductase. So the Q in complex 1 and 2 represents ubiquinone or coenzyme Q. And we're going to see in the next slide what ubiquinone does. So basically, NAD gives its electron to ubiquinone, and succinate also gives, succinate reductase also gives its electron to coenzyme Q, which is what it represents here. We have complex 3, which is cytochrome C oxidoreductase, and it has other subcomponents. We have complex 4, which is cytochrome C oxidase, and it also has its subcomponents. All right, I mentioned before that we're going to see what Q does. Q, also called coenzyme Q or ubiquinone, and cytochrome C are what we call mobile electron carriers. Mobile, when you're mobile, it means you can move or you move a lot. So in our electron transport chain, electrons are moving from one complex to the other. But those complexes we are going to see are they're attached to the membrane of the inner mitochondrial membrane, and they cannot move. So they need other molecules to help move those electrons from one complex to the other. And ubiquinone is one of those mobile carriers. Cytochrome C is also one of them. We are saying that in the electron transport chain, we are moving electrons. Where did these electrons come from, or where do these electrons come from? We have seen in most of the pathways that we have seen before, well, just glycolysis and the TCA cycle, we saw that some of those steps involved the release or the use of NAD. If you remember what NAD is or FAD, please write in the comment section the full meaning of that. But most of the pathways or most of the reactions that are catalyzed by dehydrogenases would release NADH, the reduced NAD, the NAD that has electrons, which will go into the electron transport chain. Or some of them will produce FAD, like the succinate dehydrogenase that we know in the TCA cycle. So these are the source of the electrons that go into the electron transport chain to eventually produce energy for the cell. So we can basically call NAD and FAD cell batteries, the batteries of the human cell, because they store energy in the form of electrons. Not like they have the energy, but they have what will be able to provide energy for the cell. All right, so here's a diagram just trying to illustrate what we have talked about before, the place where electron transport chain occurs and the different components of the electron transport chain. Here you can see the complex 1, we see complex 2 here, and we see Q. The reason why we're seeing Q attached to complex 1 and 2 because they give their electrons to Q, coenzyme Q or ubiquinone if you call it that. And then we have complex 3, we have cytochrome C here, it's another mobile carrier, can move. So Q will move electrons to complex 3, and then cytochrome C will move electrons to complex 4. So these are the different complexes, and all of them are in the business of receiving and transferring electrons till the last receiver of electrons, which is oxygen. And then we will see something interesting here. This structure here is ATP synthase, the enzyme. So this electron transfer does not just randomly produce ATP from space. At the end of the day, something in machinery, like the turbines that we're talking of in the electrical system or in the electrical power plant, is going to also turn to produce ATP for the body. All right, this slide just shows you another uh, arrangement of the different components of the electron transport chain. Remember, they are all arranged on the inner mitochondrial membrane. And complex 1, complex 2, all giving their electrons again to coenzyme Q. Coenzyme Q is a mobile carrier, carries it to complex 3. Also transfers the electrons through its components to cytochrome C, which is a mobile carrier. And then eventually we have movement to complex 4 and the last electron acceptor, which is oxygen. So we are going to see this, but because it's present in this diagram, I just want to let you know that when electrons are moving, something else is happening, the movement of protons. Now, when in chemistry, biochemistry, you should look at the direction of arrows when reactions are involved. We see that the arrows are showing outside. So it's moving out of the matrix into the intermembrane space. We're going to see it. Protons are moving out into the intermembrane space, while electrons are moving through complexes till the last one. The electron transport chain is based on a very interesting theory called the chemi-osmotic theory. This theory explains basically how oxidative phosphorylation occurs. And the theory states that the free energy that is released during transport of electrons through the electron transport chain is used to translocate protons actively from matrix to intermembrane space of the mitochondria. So I want us to highlight some things here. Free energy, transport of electrons, translocation of protons actively into the intermembrane space. This is basically what I was explaining in the previous diagram, the movement of protons. We're going to see it better in the next slide. During transfer of electrons, free energy is liberated. So when complex 1 brings its electron, complex 2 brings its electron, and those electrons are moving, there's always protons released. The energy that is released, the free energy that is liberated during the transfer of those electrons, causes translocation. When you translocate, you move from one location to another. So it causes translocation of protons from the matrix to the intermembrane space. So let's see it in the next diagram. Okay, so remember I told you to take note of where the matrix is, where the intermembrane space is. So this is the inner mitochondrial membrane where all the complexes are found. This is the intermembrane space because it's a space between the inner and the outer mitochondrial membrane. And the matrix is the inside of the mitochondria. So the complexes are arranged on the inner mitochondrial membrane, and electrons come from the electron batteries or cell batteries that we saw. And those electrons move from complex to complex. So there's a transfer of electrons. In the chemi-osmotic theory, the free energy that is released when you're moving electrons from here to here is used to translocate protons. Where are the protons? They are located inside here. If you are translocating them, meaning you're removing them from their original location to another location. So the free energy used or released, when electrons are moving from one complex to the next is used to power the movement or the translocation of protons from this matrix to the intermembrane space. And we're going to see that not all the complexes cause translocation of protons. We see that all of them do except complex 2. So when complex 1 is moving electrons, protons are translocated. It produces enough free energy to translocate protons. However, complex 2 does not release a lot of free energy that can translocate protons. And then complex 3 does the same. Complex 4 also does the same. But the number of protons released are also different. So that is a chemi-osmotic theory. It is what drives ATP synthesis at the end. We're going to see that. So I hope you understood this theory. Okay. The chemi-osmotic theory describes, as we have said before, explains how the electron transport chain or oxidative phosphorylation occurs. So oxidative phosphorylation from its name is addition of phosphate, and we know that ATP, which is the energy currency, has phosphate. So we have not yet explained that side of the story. So we are still talking about the fact that protons are translocated from the matrix into the intermembrane space. Sorry. So this is where we get to see the turbine that we talked about at the end when we're talking of the structure of, or when we're looking at the position of the different complexes on the inner mitochondrial membrane. So now, in continuation of this chemiosmosis, what is the use of the cell trying to pump protons into the intermembrane space? The protons are pumped into the intermembrane space during transfer of electrons from one complex to the other. And what happens is that at the end, there is a proton gradient. When you're talking of a gradient, you're usually talking of different or varying concentrations of something. When you talk of concentration gradient, for example. So causing a proton gradient means that since protons have been pumped from the matrix into the intermembrane space, so what happens? Those protons become higher in the intermembrane space. As you can see here, there's a high concentration. There's a high concentration of protons in the intermembrane space. This is what is going to cause ATP to be produced. Because think about it, the body is trying to produce ATP from scratch. But as we have seen with most of cell pathways, the cell does not produce energy without also using energy. So somehow energy has to come from somewhere. So we are trying to produce ATP here. We will not use energy from ATP again. There's another way of producing energy. And the body does this, or the cell will do this by creating this gradient. So the gradient has been created. There's a higher concentration of protons here, but the body does not also like when there is no balance. So it will try to balance it again by making those protons to move back into the matrix. But now the protons will not move back in through the complexes that they came out from. The protons will pass through ATP synthase. So this structure here is ATP synthase. Remember, we have talked of enzymes before, we have mentioned enzymes before, and we said syntheses are enzymes that synthesize something or that produce or manufacture something. So ATP synthase is the enzyme that manufactures ATP. That's why the electron transport chain is the the power plants of the body because it produces ATP from scratch. And if you're manufacturing it from scratch, it will definitely be in larger numbers. So now, what happens? The proton gradient was created. Now the gradient causes one side to have a higher concentration than the other side. The body does not like balance, so we try to balance it. The protons will move back inside, but they are moving back through ATP synthase. So the proton has to move back through ATP synthase. And when moving through ATP synthase, that's what powers the movement or the turning of this turbine, our bodies or the cell's turbine, which is ATP synthase. So protons moving in back to the matrix through ATP synthase causes the turning of ATP. You can see the direction here. There's a movement, a cylindrical rotor that moves. At the end of the day, the turning converts ADP, or it adds it adds phosphate to ADP to create ATP. So this ATP synthase is the enzyme that will produce ATP. So this form of phosphorylation or production of ATP in the electron transport chain is different from the ones we saw in TCA cycle, for example, where we call substrate level phosphorylation. This one is oxidative phosphorylation. This is oxidative phosphorylation and it is production of ATP from scratch with the use of ATP synthase. In substrate level phosphorylation, we see first there's a different enzyme being produced there, and it happens only at the level of that step. And not much ATP is produced, just one ATP is produced. Now, as we already said, ATP synthase is a complex. It's a complex which has part of it, the F1 and the F0 complex, which are located in the inner mitochondrial membrane. Of course, we have seen that. And we have already mentioned that when protons are passing or re-entering the matrix, they pass through the F0 complex and results in a change here. So this is the F0 component or subcomplex of the ATP synthase, and this is the F1 component of it. And when protons are moving into the matrix, they pass through this F0 complex, and this part, the F1 complex, is what turns to produce ATP from ATP. Okay, so I hope we have been able to understand oxidative phosphorylation. So basically, oxidative phosphorylation basically just describes the formation of ATP from ADP and P at the level of the ATP synthase mechanism that we have just described. The first part of it is able to explain how energy is generated to produce that ATP, to produce the ATP through ATP synthase. Basically, the oxidative phosphorylation is just the part where ATP is produced through ATP synthase at the end there, but we have said that this cannot happen without the electron transport, and it cannot happen without the translocation of protons into the intermembrane space. So oxidative phosphorylation couples with the chemiosmotic theory that we, or chemiosmosis that we have described before, and chemiosmosis happens or is coupled with electron transport.